So as a brief outline of what I'm going to talk with you about today, first I'm going to introduce some of the different anatomic sites that are available for blood collection in mice. And I'll then discuss some of the factors that affect the impact that these particular phlebotomy methods can have on a mouse and ways that we can assess or quantify the impact of phlebotomy on mice. I'll then talk about how your phlebotomy site choice as an experimenter can actually impact your study data. And I'll then discuss how the phlebotomy choice can actually impact the performer themselves and how the performer's skill can potentially differentially impact the success of the different phlebotomy techniques. I'll then talk about some of the difficulties that exist when we're trying to compare published studies that have evaluated phlebotomy methods in rodents. And I'll take a brief look at some of the published data that's out there for looking at phlebotomy techniques in mice. And this is just a few of the papers. There's many more out there. I just selected some I thought would be interesting, but didn't try to make a value judgment on which ones were included or excluded. And then I'll talk about a paper that we recently published at Cornell. It came in JLAS last year, where we evaluated the impact of a modified tail-tip amputation method for collecting blood in mice. And just some learning objectives to keep in mind throughout the presentation. I think it would be great to think about ways that are things that you need to actually consider when you're selecting an appropriate phlebotomy technique. So when you're designing a study, think about how your selection of the phlebotomy technique can impact your animals as well as your data and the people that are collecting that data. Also consider some of the factors that are associated with a certain phlebotomy technique other than just the site of location that you choose to collect blood from that can determine the impact that that method will have on the animal's perceived pain and distress. Also to consider the methods that we have available for assessing the impact of that technique on the mice. And just be aware of some of the difficulties that exist as we try to interpret the published literature that's out there that's describing these various phlebotomy techniques in rodents. So as I mentioned, there are multiple sites that exist in the mouse from which an experimenter can collect blood. Some of the more commonly used I have depicted here. So lots of researchers will use the submandibular vein. It's sometimes called the facial vein. The retrobulbar plexus or sinus is commonly used in rats and mice. Sometimes this will be called retroorbital in the literature. The lateral tail vein is also frequently used, as well as the saphenous vein for blood collection. You can also get blood from mice from the jugular vein, actually from the sublingual vein, and from tail-tip amputation. And there's some other techniques that I won't have time to mention today, such as superficial temporal blood collection, but these are the main techniques that I'll discuss at least briefly today. So most of my talk is going to focus on the impact that the phlebotomy technique will have on the animal, but it's also important to realize that the impact, that the phlebotomy method will also impact your data and potentially impact the performer. And since recommendations for phlebotomy techniques that are proposed either in working group reports, individual literature papers, or even developed in-house vary, a researcher really needs to kind of do their own evaluation and consider the impacts that the phlebotomy method will have. So now let's talk about some of the major factors that will influence the degree of impact that a phlebotomy method will have on the mouse. So primarily, I think of restraint and handling as also having a large impact on the perceived pain or distress by the animal, in addition to the actual blood sampling procedure itself. And so restraint varies greatly among the different techniques, ranging from full physical restraint, such as a scruff or the use of a restraint device, to minimal restraint, such as just holding the base of the tail. And some methods actually also require anesthesia. The length of time that's required for the different blood collection methods to get the sample of blood that you need will also vary. So blood flows at different rates from the different sites. It's going to really depend on the size of the vessel that's punctured as well as the blood pressure at that site. And the length of time is probably going to have a big impact on certain parameters that you might measure, particularly if they're impacted by a stress response. And if that's the case, then you would hopefully get your desired sample collected before you would have activation of the hypothalamic pituitary adrenal axis, which is what's going to be responsible for changing those stress responsive hormones. You should also think about the innervation at the site that will be punctured. So some areas are going to be more highly innervated than others, and that's going to impact the acute pain response that's perceived by the mice. I also think it's important to consider the physical use of the anatomic site that's going to be punctured. So for instance, with the submandibular or the facial vein phlebotomy, if that causes tissue damage to the mascatory muscles as the mice are chewing post-procedure, they might experience discomfort. You could also think about the saphenous vein. If there's some tissue trauma in the leg, that might cause discomfort during ambulation. So it's important to think what the mice might also use that site for. It's also important to consider the risk of secondary injury. So we'll continue to talk about this as we mention these specific phlebotomy options, but there are a lot of reports in the literature that using the retroorbital or retrobulbar method has significant risk for injury to the globe, to the Harderian gland. As kind of I just mentioned, using the submandibular or the facial vein, that site's very close to other important anatomic structures such as the ear canal, the temporomandibular joint, the facial nerve, the mascatory muscles. So just thinking about what is the risk for potential injuries to other structures, particularly if the first attempt at the phlebotomy method might have gone off target. And it's also important to consider how the preparation of the site can impact the animal. Even for the same anatomic site, you'll read that reports vary in how they prepare the site. So sometimes when you're reading a report that's looked at tail vein incision or tail vein puncture, they might mention warming the tail ahead of time for vasodilation. Some techniques that describe saphenous venipuncture, they shave the site or they apply alcohol, and others do no prep. So the amount of just manipulation of that area might also impact how the animal perceives the stress of the procedure. So now that we've talked about some of the things that can impact how the animal perceives the procedure, even independent of the location, let's talk about some of the ways that we can actually assess as an investigator or a researcher the impact that the method we could choose would have on the animal. So I've divided these into looking at physiological parameters and behavioral parameters. And it's important to measure more than one parameter. And I also wanted to make a note that we don't really have a way to weight the relative importance of any changes that we might find. So you might find that one technique causes a change in parameter A but not B, and another technique causes a change in parameter B but not A, and we don't always know which one is more important to the mouse if there's a change in parameter A or B. So I think that's also a reason why it's important to look at multiple parameters. So when we think of physiological parameters that we can measure, a list of some I've come up with here could include, measuring body weight or food consumption. You could look at reproductive performance, especially if you were doing some sort of serial blood collection, and these were in breeding mice. Looking at vitals could be very informative, but it may not be very easy to do unless the mice have a telemetry device. Often you'll see in papers people have measured plasma corticosterone, and that's because it's the major corticosteroid in mice. I did want to point out that if you measure corticosterone in plasma, that's letting you know sort of about a narrow window of physiologic time. There's other options. You can also measure corticosterone in feces or hair, and that could give you a better integration of stress levels potentially over time. We can also measure blood glucose. That's a really quick and pretty inexpensive measurement. But hematologic parameters and histology changes can also be used to assess the physical impact of a technique on mice. And then we can also use behavioral parameters to study the impact of a phlebotomy method on mice. And so two of the more common ones you'll see, if you look in the literature, are the open field test or the elevated plus maze. And these are both two unconditioned response tests. And they evaluate aspects of natural rodent behavior, such as activity and exploratory behavior, as well as aversion to the open areas. And we can also look at nesting behavior and grooming, just their general activity, their posture. And these things are great because they can be done sometimes without even moving or disturbing the cage at all. And you can also potentially record vocalization. So as I mentioned, for most of this presentation, I'm going to focus on the impact that the procedure has on the animal. But I did think it was important to just point out that the method that you choose to collect your blood sample can also impact your data. And so the types of physiologic responses that the method can trigger in the mouse can actually lead to some pre-analytical variation in your data by changing some of those physiologic parameters that we just talked about, such as corticosterone, glucose, free fatty acids, and the leukogram. And as I mentioned before, if you're worried about these, it would be best if you can get your full sample collected before you've had time to activate the hypothalamic pituitary adrenal axis, which generally takes several minutes in rodents. And so then a method that required, minimal restraint, minimal handling, could achieve the blood collected, the desired volume in a short amount of time, and hopefully did not require any administration of analgesia or anesthesia. That's kind of its own procedure. Those might be more favorable if you're trying to measure these stress-responsive parameters and potentially mean that you're going to have less variability in your data. And so just to give you an example of this, in the literature, there are several reports of how the phlebotomy method can impact the actual data that you collect from your blood sample. So in a report by Christensen et al., they showed that if you collected blood from the tail tip, amputation or puncture, you had quite a bit of hemolysis in your sample, and that can change some of your chemistry values. But they did not find hemolysis if the blood was collected through retroorbital bleeding. In a paper by Asland et al., they showed that you would get a difference in your glucose sample whether or not you took blood from the tail vein versus the saphenous vein. But they didn't find actually any differences in hemolysis from blood collected at those two sites. Abaten et al. Showed that hematology white blood cell parameters can be different based on whether or not the blood was collected from the saphenous vein, where the white blood cell counts were lower than if they were collected from the tail tip. And Fernandez et al. Showed that multiple chemistry parameters can be different whether or not blood was obtained from the submandibular vein versus the retroorbital plexus. So these are just a few examples. And as I mentioned, for most of the talk, we'll talk about how the phlebotomy choice will impact the animal. But it is also important to know that it could impact your data, and maybe you should do a pilot study first just to make sure that that sample collection site will be what's most optimal for the parameters that you're going to measure. I also wanted to touch briefly, before we move into more about the impact on the animal, about how your phlebotomy choice can impact the people that are responsible for collecting the data, and how that person also can impact the success of the method. So certainly the level that's, of stress that's perceived by the person who's responsible for collecting these very precious samples can be different based on the technique that they're expected to master and be successful at. And the different phlebotomy techniques, they vary in how feasible they are and the amount of training required. So some of these techniques require, using just anatomic landmarks to do a blind puncture where you can't see the vessel that's your target. Some of them require maybe holding the animal in a more difficult position. And so some of these can be more or less stressful as perceived by the person who's going to have to be actually doing this. And on the flip side, the skill of the performer can also impact the success of the phlebotomy method differentially based on method. So what I mean by that, I'll show you an example here. There's several reports like this in the literature. But this paper by Abaten et al. Compared the impact of the expertise of the performer on plasma corticosterone response in using two different methods. So they looked at saphenous venipuncture and tail tip amputation. And so here on the left in A, we're looking at saphenous venipuncture, and you can see that the plasma corticosterone was significantly higher in mice that had blood drawn from a novice phlebotomist versus mice that had blood drawn from an experienced phlebotomist. And in contrast, on the right in B, you can see that there was no statistically significant difference in the plasma corticosterone of mice that had blood collected via novice or experienced phlebotomists via the tail tip technique. So this suggests that at least the animal's perceived stress response with blood was collected via the saphenous puncture was dependent on the skill of the phlebotomist, but not in the mice that had blood drawn from the tail tip amputation. So potentially, you can kind of get away with a little bit less skill and not have an impact on the perceived stress by the mice with some techniques than others, which makes sense if some techniques are just harder to perform and be successful at than others. So now that we've discussed how the phlebotomy method can impact the study animal and the data, as well as the performer and some of the ways that we can assess the impact on the animal, let's now talk about some of the difficulties that exist as we're trying to read and interpret the literature that's out there that's looked at phlebotomy methods in mice. There are several difficulties that exist when you're trying as someone who's interested in either animal welfare or just picking the right correct or the most appropriate phlebotomy method for your study, and you're out there reading these papers. And the main one I've already touched on, and that's that techniques differ for blood collection even from the same site. So as I mentioned, for tail vein incision, there'll be some reports that talk about using a restraint device, heating the tail ahead of time. Sometimes the blood is collected over up to 8 minutes, I found in one report, by tail incision, versus others would use tail incision with very minimal restraint and get the blood in a much quicker fashion. Some people will use anesthesia for the same technique that others wouldn't. And so this makes it very difficult when you want to compare the impact across studies if the whole sort of preparation and procedure of the animals varies so much. When researchers design these studies to assess the impact on animal welfare of these different blood collection techniques, they generally have to choose whether they want to control for either the volume of blood that they collect, which could impact the mouse, or the amount of time it takes to get the blood sample. And we mentioned this before, blood's going to flow at different rates from the different anatomic sites, so it's very difficult to control for both. So you need to keep that into account. Sometimes, you might get a whole lot less blood from one mouse than another in the same amount of time, and the investigators cannot usually control for both. I also noticed when I was trying to do a literature search on these phlebotomy method papers that often one phlebotomy technique would be compared to another or to several others without the use of an actual control or sham, a handled only group. And I think it's important to consider if you're going to design an experiment that evaluates the impact of a phlebotomy method on mice, to include a sham group, because that provides potentially valuable information regarding the potential benefit or lack of a benefit of providing anesthesia or analgesia. So if your phlebotomy group doesn't behave any differently in the physiologic and behavioral metrics of pain or distress that you measure than a sham-only group, then that likely suggests that providing analgesia or anesthesia is not really going to offer a welfare benefit in that case. Okay, so now that we've discussed some of the difficulties in trying to glean information from the studies that are out there on phlebotomy, I'm just going to talk about some of the main common methods and give some of the very broad general characteristics about these common techniques, and then I'll go into some of the select studies that evaluated them. So retrobulbar, or often I said it's referred to as retroorbital phlebotomy technique, it generally is described as using a capillary tube to go at either the medial or lateral canthus of the eye. I've seen both reported. Some institutions will require that mice be anesthetized for this procedure. You can get large volumes of blood quite quickly using this procedure. Some institutions will restrict the number of serial samples that can be collected from the same eye. There is potential for damage to the globe and the Harderian gland, and that's well documented in the literature, and is usually cited as one of the main disadvantages of this technique. There is a potential for excessive hemorrhage, and this method just is not always pleasing aesthetically to some performers. The lateral tail vein is another very common technique, and as I've mentioned before, the restraint can vary. So in this picture, you can see they're employing a restraint device. Other people will just minimally hold sort of the base of the tail and allow the mouse to have kind of freedom of movement. You can achieve large volumes with a lateral tail vein, but it will take a little bit longer, and often you're going to sort of be milking the tail and kind of collecting drop by drop. It can be performed serially, and if the serial samples are collected close enough together in time, you can often just disrupt the clot to still be able to collect a sample. This method has the advantage that it's not near other important anatomic structures or tissues, and the tail vein is often visible. It may be a little harder to see in pigmented mice, And if there's any excessive bleeding, you would be able to see that as well. The saphenous vein is also reported in the literature. You can see varied ways of holding the mice. Some people will actually just hold them in their hands. Other people do use a restraint device, but usually they'll describe performing some kind of vessel occlusion proximal to the site you want to collect the sample. You can get large volumes from the saphenous relatively quickly. It can be used for serial blood collection. It is reported that saphenous vein collection can cause tissue trauma at the site. It does have the advantage, though, that the target is visible. And then papers will vary in how they describe the preparation of the site, as I've mentioned before. So some papers describe shaving, others don't, or maybe applying alcohol to help you see the vein. But obviously those might impact the level of stress that's going to be perceived by the animal. The submandibular vein collection is often called facial vein. I might use them interchangeably. It's also commonly done. It does require full restraint, which is usually just manual restraint with scruffing, like what you can see in the picture. There are some reports, though, that actually recommend anesthesia, while others say that anesthesia provides no improvement in welfare indicators. You can definitely get large volumes from the submandibular vein quickly, and it can be done serially. But as I've mentioned a few times, it is near other very important structures in the face. So, you're close to the facial nerve, the ear canal, the temporomandibular joint, those masticatory muscles, and their salivary glands and lymph nodes sort of all in that same area. One of the main disadvantages, I think, is that you're relying on anatomical landmarks and you don't have a visible target for what you're trying to puncture. And so you can get severe histologic damage, and that's in several independent reports. And you can also get blood from the jugular vein in mice, and reports vary. Some describe doing this in awake mice with full physical restraint, and other reports describe doing this in anesthetized mice. You can get large volumes from the jugular, again, very quickly, and it can be used serially, but there is a potential for damage to surrounding tissue and the potential for hemorrhage. So now that I've talked about some of these common techniques, I'm just going to present a paper that actually compared these 5 common techniques by Cyano et al. That came out in a 2015 lab animal paper. And as I go through some of the literature that I've selected to present today, I've made a table so I can try to give you information regarding the type of restraint that was used, the instrument that was used, whether or not they prepped the site, how long it took them to collect their sample, the volume of the sample that they took, and any information they provide on whether or not serial samples were collected. Because I do think as you're reading these papers, it's really important to try to pull that out. So in this 2015 paper that I mentioned, they compared retrobulbar phlebotomy either with a thick capillary tube in awake mice or retrobulbar phlebotomy using a thin capillary tube in mice that were actually anesthetized, to tail vein collection in which they used a restraint device, saphenous vein collection, again, in which they used a restraint device, to submandibular vein where they scruffed the mice, so manual restraint, and as well they also included a jugular vein group, and those mice were anesthetized for jugular vein phlebotomy. So I've listed here the instruments that they have used. Also note they did prepare the tail vein group by putting their cage under a red light for 10 minutes. In this paper, they chose to control for the volume of blood they collected. So they collected approximately 75 microliters for all of the techniques. And if you look at the duration of time, they were actually great in reporting the duration of the entire procedure, which was, from removing that mouse from its cage, and also just the length of time it took to collect the blood alone. So you can see that the retrobulbar method in awake mice was the shortest length of the procedure, and if we look at the anesthetized procedures, those took quite a bit longer. So what they did to actually then measure corticosterone, they took blood from one mouse from each cage for 6 days via the retrobulbar technique, and they averaged the corticosterone value, and they call that their baseline. And then they performed this phlebotomy technique, these 6 listed here, 15 minutes after they performed that technique, they went back and drew a second sample through the retrobulbar method. And it's that sample in which they measured the corticosterone. So the data I'm going to show you about corticosterone didn't actually come from blood that was collected by one of these 6 techniques, but rather from blood that was 15 minutes after that technique. So hopefully then able to assess the stress response post-hypothalamic pituitary adrenal axis. So they gave that time to kick in, and then they tried to measure the stress response associated with these 6 different techniques. So this paper by Cyano et al. Showed that retrobulbar phlebotomy may actually cause less physiologic and behavioral changes than the facial vein or submandibular vein, saphenous tail vein, and the anesthetized jug response, jug phlebotomy. So as I mentioned, they measured plasma corticosterone, and I'm showing you that data here for all of the groups. So the baseline, that sample that was taken an average for over 6 days, is in the red. And then 15 minutes after bleeding, by the experimental techniques, they took a sample and measured the corticosterone. So you can see that both retrobulbar A and B had sort of lower increases in plasma corticosterone than some of the other techniques. But it is important to note that all the techniques did cause a significant increase in plasma corticosterone relative to baseline. They also came up with a scoring system, which I've shown you here, for in-cage activity after the experimental phlebotomy technique. So with increasing scores with increasing activity, and I'm showing you this data here. And so you can see that retrobulbar phlebotomy was associated with a high in-cage activity after the procedure, which is interesting even in the retrobulbar group that had received anesthesia. And then I'm not showing you the data, but this Cyano et al. Paper also reports that retrobulbar phlebotomized mice had a moderate speed and distance traveled in the open field. And another really interesting thing that this study did was compare the anesthetized versus the non-anesthetized versions of doing a retrobulbar phlebotomy technique. And they show that anesthesia was actually associated with a greater increase in corticosterone, which you can see again in the data there on the left. This study also compared using the thin capillary tubes versus the thick, and they showed that in the retrobulbar phlebotomy performed with the thin capillary tubes, they actually saw less histologic changes at the site than they did in the groups that had saphenous tail vein or facial vein phlebotomy. So how they did this, they looked at 8 indicators of histologic changes. So I've listed those here, things like hemorrhage, inflammatory cells, degenerative changes to the tissue, edema. And they looked in multiple tissue types. And so they calculated this incidence of histologic changes, and they provided a percent of the total possible score. So in their scheme, there were 128 possible for the non-retrobulbar groups and 192 points possible for the retrobulbar groups. And they looked 1 hour after phlebotomy, as well as 3 days and 14 days after phlebotomy, and you can see that the retrobulbar phlebotomy groups, particularly retrobulbar group B that had the thin capillary tubes, have the lowest incidence of histologic changes 1 hour after, as well as low histologic changes 3 days after. But it is interesting to note that it seems like potentially the histology changes at the tail vein sites in the tail vein group may resolve quite quickly. So there was kind of a steep drop in the percentage of changes in that group from 1 hour after to 3 hours after tail vein phlebotomy. So this Cyano et al. Paper found that lateral tail vein phlebotomy sort of had intermediate impacts on physiologic and behavioral responses. So mice that had tail vein phlebotomy had a large increase in corticosterone, but they also had high in-cage activity and high speed and a large distance traveled in the open field. Similarly, they showed that the saphenous vein phlebotomy also seemed to have kind of a rather intermediate impact on these metrics that they measured. So the saphenous vein phlebotomy group had a pretty low increase in corticosterone. As you can see, it's the fourth group here on the left. But the saphenous vein group had a high in-cage activity, which suggests minimal impact, and a high speed and large distance traveled in the open field. The same paper, we're still talking about the Cyano paper, showed that submandibular vein phlebotomy was associated with an increased incidence of physiologic and behavioral responses versus the retrobulbar and the tail and the saphenous. So the submandibular, or they call it the facial group, so it's abbreviated FVB, was associated with large increases in corticosterone. So it's the second to last group here. And so that increase in corticosterone was very similar to the anesthetized jugular vein group and higher than the other phlebotomy methods. And mice that had the facial vein phlebotomy also had decreased activity post-phlebotomy, and they tended to move short distances and move slowly in the open field, performing, again, similarly to the anesthetized jugular group. So we kind of just talked about the anesthetized jugular group was pretty similar to the facial group, but they did have the greatest increase in corticosterone, and again, low in-cage activity, low speed and distance traveled in the open field. But I did want to point out for fairness that the impact of saphenous vein phlebotomy that's reported in the literature, it does vary by report. So similar to this sort of Cyano paper where we found sort of this intermediate impact of saphenous vein phlebotomy, Aslan et al. Showed that there was lower stress response after saphenous venipuncture than after tail vein blood collection. Avitan et al. Showed that plasma corticosterone did not differ over 4 samples that were taken in 1 week by the saphenous vein. But there are reports, such as the Matatoja report, that show that there's higher stress after saphenous venipuncture than after facial vein or tail vein. So again, this kind of goes back to what we talked about with some of the difficulties in carrying these techniques. This really may depend on whether or not the animals were restrained a certain way, how long it took to do the procedure, and whether or not there was any particular preparation of the site. So just, there's other reports that also evaluate the saphenous vein, but just to kind of, I wanted to make sure that you were aware that the impact of saphenous vein kind of depends on what paper that you read. Similarly, the impacts that are associated with jugular vein phlebotomy can vary by study. So in the Cyano paper that we just looked at, they had anesthetized the mice for jugular vein collection, and those mice had, with facial vein, some of the most kind of severe changes in those metrics that they measured. But there is a paper by Shirasaki et al. I think the date got a little cut off. It's supposed to be 2012. And they actually performed serial sampling from the jugular vein, and they showed that it had less impact on serologic markers of physiologic stress versus the lateral tail vein. So they compared awake jugular vein collection with lateral tail vein collection that was done with mice in a restraint device. And then they looked at C-reactive protein, and they showed that actually the lateral tail vein group had higher C-reactive protein, which can be a marker of stress, than the mice that had blood collected from the jugular vein, and that the 2 groups had similar plasma corticosterone. So again, just kind of this, if you only read this paper, you might get a different view of jugular vein phlebotomy than if you had only read the Cyano et al. Paper. So it is important to kind of think about the whole technique of the method that is described. This Shirasaki et al. Paper, they showed that even with the serial sampling done once a day for 5 days, in their words, that there were only slight hematomas and damage to the surrounding tissue, and they show the image of that here. So now moving on to a different technique that's not used as commonly, and that's the sublingual vein collection. So there are 2 veins that are running from the basis to the apex, and unlike in some other species, they're only thick enough for venipuncture actually near the base. And that's different from in rats and in hamsters, where you could puncture a larger portion of that vessel's length. So sublingual collection is performed with full restraint. Some reports talk about using anesthesia. Sometimes it requires 2 performers, so one person would sort of be holding the mouth, another extracting the tongue and performing the venipuncture. You can get large volumes of blood this way relatively quickly. There is a potential risk that the mouse could aspirate any blood that's left in the oral cavity, and there's also the potential that that blood sample is going to get mixed with saliva. So saliva contains several enzymes that actually start the digestive process. So you might be worried about that as far as sample quality. But there was at least one paper I found that looked that sublingual blood collection, at least in rats, did not seem to impact amylase activity in the blood sample that was collected. So potentially that's not a huge concern. But one of the things that I think I would probably be at least interested in is whether or not sublingual collection could impact food intake in mice. And that was actually looked at by Hyman et al. In 2010. And they found that sublingual vein phlebotomy was associated with no change in food consumption or weight loss versus submandibular phlebotomy. So they looked at either Group 1, where mice had the submandibular vein punctured, and those mice were just manually restrained. Group 2 were mice that had the submandibular vein punctured, but they were actually fully anesthetized. And then in Group 3 were mice that were anesthetized for sublingual collection. And then they looked at food consumption, and you can see that actually, so they measured several days before the phlebotomy, and then you can see where they've indicated they've done the blood collection and then measured food consumption for several days after. And you can see that Group 3, which was actually the sublingual vein collection, had higher food consumption than the two groups that had submandibular phlebotomy. They also looked at histologic changes, and so this is a bit of a busy graph, but again, they're divided by the groups. So Groups 1 and 2 were the submandibular collections, and Group 3, the sublingual collection. And then as the row titles here on the left, you can see the types of histologic changes that they were identifying. And there were a greater percentage of mice, as well as having more severe histologic changes in the submandibular group versus mice that had the sublingual vein collection. And so then I'll move to the final technique that I'm going to talk about today, which is modified tail tip amputation. So this technique was first reported by Abbatan et al. In 2008. And this technique, they reported performing it with minimal restraint, so just holding the base of the tail. You can collect large samples of blood this way, but it will take longer than some of the other methods. So similar to tail vein incision, you know, you're going to kind of be milking the tail and sort of collecting drop by drop. As some tissue is actually removed, it's likely not suitable for serial collection in which the samples are collected over a broad span of time. However, if you can just disrupt the clot, that might be just fine. It has the advantage that there's no damage to surrounding structures and that the target is very visible and there's very minimal excessive bleeding. And so we were interested in this technique because we had an investigator here who wanted to measure several stress-responsive parameters, and she was very interested in using a technique that required very minimal handling and minimal restraint. So we did perform a study here. Actually, first let me talk to you a little bit, sorry, I got ahead, of what actually was reported in the original paper by Abbatan et al. And they showed that tail tip phlebotomy was associated with fewer behavioral responses and a similar corticosteroid response as saphenous venipuncture. So here are the details of how they performed their study. They looked at that modified tail clip with minimal restraint to saphenous collection when mice were placed in a restraint device. And then they looked at these acute behavioral responses, such as pulling, flinching, vocalizing. And you can see that the saphenous group, which is on the left column, had a higher incidence of these acute behavioral responses than the tail clip group. And then they also measured, as I mentioned, corticosterone, and they saw that the two groups, the saphenous and the tail clip, had very similar plasma corticosterone. So as I mentioned, we were interested in assessing this modified tail clip impact on mice compared to two other techniques that were commonly used. And one of the other reasons we were interested in this is because most institutions have a policy that regulates tail snipping for genotyping. So many institutions, including ours, will require either analgesia or anesthesia if you're going to remove the tail tip for genotyping in mice over 21 days of age. And we thought it was pretty important to point out that it is a different technique. So usually people are removing several, up to 5 millimeters of tail for genotyping, versus we were just removing, as we can kind of see in the image, the lower image here, just kind of the very, very, very distal 1 or 2 millimeters. So we thought it might be important to actually evaluate the impact of this technique on mice. So we looked at either groups that had blood collected with submandibular or facial vein, is how we called it in our study, puncture, and they were manually restrained. We also had a tail vein incision group, and then our tail tip amputation group. And then we included 2 sham groups. So the sham submandibular group, they were scruffed and handled just like the facial vein group, and we also had a sham tail group that was minimally restrained on the top of the cage and had the tail kind of milked, even though there was no actual puncture. And we chose to limit the length of time for the procedures. So the submandibular or facial vein group was actually, it was much quicker to obtain the maximum volume of blood allowed. So we went with the by weight calculation. But then for the tail groups and the sham groups, we timed it out at 2 minutes of manipulation. So just briefly, the timeline of our study. During the procedure, we recorded any audible or ultrasonic vocalizations, and we also noted acute behaviors. And we then placed mice into a cage for 10 minutes and recorded behaviors such as activity or inactivity and grooming. Then mice went into an open field test for 5 minutes. They then had 5 minutes just sort of as a rest period back in that observation cage, and then they were placed in an elevated plus maze for 5 minutes, and both of those behavioral tests were video recorded. Mice then went back to their home cage, but 6 hours after phlebotomy, we moved them to a new cage that had a brand new little cotton nest pad, and they stayed in that cage for 4 hours so we could evaluate their nest building activity. And then roughly 12 hours after phlebotomy, they were euthanized for histologic tissue evaluation. So quickly, we did find that tail tip amputation was very feasible. We found no significant differences in the volume of blood that was collected, so all of them allowed us to collect the maximum volume of blood that would be allowed. And tail tip amputation had the highest number of successful first attempts. So all 9 of those mice had success on the first attempt. 3 of 9 tail incision mice had to have the incision deepened to get adequate blood flow. And unfortunately, we had only 4 of 12 of our facial vein group mice had successful phlebotomy on the first attempt. We didn't detect any differences in blood glucose between the 3 phlebotomy methods, facial vein, abbreviated FV, tail tip amputation, abbreviated TA, and tail vein incision, abbreviated TI. And this likely reflects that we collected our full sample before activation of the hypothalamic pituitary axis, adrenal axis. We did find that there was a significant impact of the phlebotomy group on vocalization. So specifically, the facial vein and sham facial vein group mice produced more audible vocalizations. And most of the vocalizations actually were produced during handling, during scruffing. And so this led us to believe that this was largely due to the handling procedure, since it also occurred in the sham facial group, and that type of restraint is probably very stressful to the mice. We didn't have any ultrasonic vocalizations recorded during any of the procedures. So during that 10-minute observation window after the phlebotomy, we looked at behaviors such as activity and grooming, and we did find a significant increase in the epochs or periods of inactivity in our facial vein group mice. And so we define those as periods of 15 seconds or more in which the mice were largely immobile. And so we interpret this to mean that the procedure, the facial vein procedure, was either more painful or distressful to the mice. And this was similar to what was reported in that original Cyano paper that I talked to you about earlier, where they also had decreased activity in their facial vein group. I thought this was quite interesting. We did find actually that tail vein incision, but not tail tip amputation, increased tail grooming. So mice generally always sort of groom their head and body more than their tail anyway. So we analyzed sort of the tail group separately, and we looked at whether or not mice, how many times they groomed their head, their body, or their tail. And mice that had the tail incision groomed their tail more, which may actually be because there's sort of a wicking effect of the drop of blood sort of down the tail. So it could have been that more than any pain or distress or both. But importantly, the tail tip amputation group paid no more attention to their tail than the sham tail group. And then we didn't find any impact on body grooming in the facial vein group, and we included our 2 sham groups in that as well. So then in our behavioral tests, looking first at the open field test, our mice did behave normally in the open field test. So for rodents, that's spending more time on the periphery and less time out in the open. But we did note a statistically significant decrease in velocity actually in our facial vein group in the open field. Again, that sort of fits with those periods of inactivity, and it also is consistent with what the Cy et al. Paper had reported with decreased activity in the facial vein group post-phlebotomy. Mice also behaved normally for rodents in the elevated plasmase. They spent more time in the closed arms, but we didn't find any impact of phlebotomy group on elevated plasmase performance. And then we also looked at nest building. So we had a blinded score pictures of the nest the mice built in that 4-hour window that started 6 hours after phlebotomy. And they used a modified published score by Deacon et al. And you can see that there was a non-significant trend towards decreased nest complexity in our facial vein group. And although this wasn't statistically significant, it might suggest that potentially because the facial vein puncture is at the site of the masticatory muscles, maybe those mice are spending less time sort of chewing and shredding the nest. And potentially, had we done a different time point or provided different types of nesting material, a significant difference may potentially have showed up. And finally, we looked at histology differences between our groups. And we did find that the facial vein puncture resulted in the most severe histologic changes. So I'm showing you here in a more zoomed out transverse section of the head. So the area of the facial vein is indicated by the arrow. And this was in a mouse that had no significant findings. Then that box in A is sort of where we're zoomed in B, and the arrowhead is pointing to some mild subacute hemorrhage in a facial vein group mouse. And then C and D are showing you sort of progressively more severe hemorrhage in some facial vein group mice that are around some other important structures, such as the submandibular lymph node and salivary gland. You can actually even see some red blood cells that are draining into the sinuses of the lymph node in D. So we did find more incidences and more severe histologic changes in our facial vein group. We're looking in E and F at some histosections from tail vein incision mice. So in E, there were no significant changes. In F, we're seeing some mild neutrophil infiltration. And then in G and H, I'm showing you histosections from mice that had the tail tip amputation phlebotomy performed. In G, you can see that the caudal vertebra is still intact. And in H, you can see that the caudal vertebra was actually transected. And in these mice, we saw usually mild fibrin, maybe mild neutrophil infiltration, which is sort of a normal response to any kind of tissue trauma. We did note, ideally, that the caudal vertebra would not be transected, but due to the sort of overall lack of response in the behavior and physiologic parameters that we measured in the tail tip amputation group, and the lack of any difference in that tail tip amputation group and the sham tail group, we think that potentially this impact is not very significant on the mice. So in summary, we suggest that tail tip amputation is a potentially superior phlebotomy method in terms of animal welfare when suitable for your study needs. So again, in summary, we found that there was no evidence that tail tip amputation caused more pain or stress to mice than just a sham manipulation of their tail, or than 2 already approved methods in awake adult mice. We found that facial vein phlebotomy may actually be the least desirable of the 3 methods in terms of animal welfare, even though it took less total handling time. So again, facial vein phlebotomy was associated with increased audible vocalizations, increased periods of inactivity, reduced speed in the open field test, and more severe histologic change. And we found no evidence to suggest that providing analgesia or anesthesia with tail tip amputation would be beneficial, given the lack of differences between the tail tip amputation and sham groups. So I'd like to acknowledge my co-authors that were on this paper with me, as well as some individuals who were important for the data analysis step, and of course, the Cornell University Center for Animal Resources and Education, where I conducted the study as part of my residency training. And I'll put my summary slide back up, and then I'd be happy to take any questions.
Thank you so much, Elizabeth, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your questions into the box that appear on the screen and click the Send button. We'll answer as many questions as we have time for today. So let's get started. Let's see what our audience first questions are that are already coming in. Our first question is, Elizabeth, how does tail tip amputation differ from tail tip biopsy that is commonly performed for genotyping?
I think that's a great question, and as I mentioned before, that's sort of one of the reasons why we were actually interested in performing this study. So at our institution and many others, if tail tips are biopsied to collect tissue for genotyping after the mouse has been weaned, so we say after 21 days, they're required to provide analgesia and/or anesthesia. And that's essentially because at that age, in most strains, the caudal vertebrae have fully ossified, and we think that the procedure is more painful at that age than before 21 days. And so when we had investigators here at Cornell that were interested in using the modified tail tip amputation technique, we sort of had concerns as to whether or not they should provide analgesia. And but there is actually a large difference in the degree of tissue that's transected. So you can take usually up to 5 millimeters of tail for genotyping, but we really were only just taking that caudal, even 1 millimeter, 1 to 2 millimeters with the tail tip amputation. And also, usually the genotyping procedure is sort of performed with some kind of identification. So usually the mice are picked up and they're scruffed. And we're performing only this minimal handling and minimal tissue removal. So that's why we wanted to more evaluate the impact that this kind of modified and minimal tail tip amputation technique would have versus the very kind of traditional tissue removal for tail biopsy and genotyping.
Thank you so much. And I want to thank our audience members who are asking questions today and remind them that any questions that aren't answered today, Elizabeth is more than welcome to answer via email. We have one more question coming in. Why wouldn't providing analgesia be a safe default when performing a phlebotomy?
I think that is a great question, and sort of I think often we think we're doing good by doing that, and it's sort of our desire to just give the analgesia sort of as a default, just assuming that something is painful and that's going to be of a benefit. But most of the time, providing analgesia, such as giving a ketoprofen or meloxicam or some other non-steroidal anti-inflammatory, is going to be another procedural event for the mouse. So it's an additional handling procedure. It's scruffing the mouse, restraining the mouse, and then giving it a needle injection to actually provide that drug. And actually, if you're going to usually give that drug long enough to kick in, you would have to do that well enough in advance of your blood collection procedure for those drugs to really be on board. And then, unfortunately, that handling impact and the stress that causes the animal may impact your data collection, what you're measuring in your blood sample. And truly, the benefit provided by that analgesia may not outweigh the additional stress that's caused by the mouse and that additional handling and administration procedure. So that kind of goes back to where I think it's really important to have these sham-only or handled-only groups in these studies that look at the impact of phlebotomy. Because if you see no difference between a method and a sham-only group, then likely providing analgesia is not going to improve the welfare and may actually cause increased stress because now you've handled the mice twice.
Wonderful, thank you. And thank you very much for your presentation today and for your important research. Do you have any closing remarks you'd like to share with the audience before we close today?
I just thank you guys for listening. I think that this is a really important topic that we keep evaluating, especially now as we get better and better metrics to be able to evaluate our impact on mice. So I think that there's still a lot out there that we can learn. So I encourage other people to continue to conduct these well-designed studies to evaluate the impacts of phlebotomy on mice so that we can continue to just improve our techniques, both to improve our data and also to have less impact on animal welfare.